Hey, Luis.
Hi,
Hi.
how's it going?
Here it's raining again, but
But
okay. So hello everyone. I'm very happy to have here today Luis Victoria Nogales. He has a bachelor in biochemistry at the Complutense University of Madrid. Then he did the master of science in organismic evolutionary and paleobiology in the University of Bonn, and then he was a laboratory technician at the Animal Paleogenomics Lab in Munich. Now is a PhD student, and his supervisor is Dr. Caterina Dulias in the Institute of Geosystems and Bioindication. I cannot pronounce the city, this German city, so you're gonna then say it. And today, Luis is gonna present us his project that is about early European agriculture through molecular study of ancient tools and sedimentary DNA. So thank you so much, Luis. And I would say that you can write if you have questions in the chat, and then as soon when Luis has finished, you can ask him question, and I think Luis is gonna be very happy also for feedbacks and contributions. So it's the floor is yours, Luis.
Hi. Well, the university is in Braunschweig.
Very
Okay, it's
difficult to
very
pronounce.
difficult name. Yeah, I don't even know if I'm pronouncing it right anyway. Okay, so I'm gonna share my screen. Okay, let me go to the presentation mode. Exactly. So, well, as said, my PhD project is on the study of early agriculture or agricultural activities using molecular tools or ancient metagenomics and sedimentary ancient DNA as a proxy, as said, for the study of well, a Neolithic site that I will explain now. So basically, my site of study is known as La Marmota. La Marmota, I don't know if maybe is known to any one of you, is under the waters of the Lake Bracciano. Well, the Lake Bracciano is very near from Rome and has been always very important to the city historically, normally as a resource of water. As said, the site of La Marmota lies under the waters of the lake, 6 to 8 meters, no, and as we see, the site of La Marmota is characterized by its exceptional degree of preservation. And what is striking is the presence of these huge wooden pillars that may have supported, well, basically the buildings, not that these people would have built, which is suggested that would have had really high dimensions, no, as we see here, 6 meter wide and 8 to 10 meters long. Also, what is important or famous for the site of La Marmota is the preservation of its good work. No, the most famous artifacts, maybe the canoes of La Marmota. Right now, there is a very intense debate which they were using this to for the lake, no, for transporting through the lake, or even there are some people who say that they may have gone to the sea with them, no. Also, we have a lot of pottery, a lot of bikestry, etc. But what is more important to me, because it's one of my, let's say, one of the, or half of, at least half of my PhD project, is the study of the sickles, no? Well, as you may know, sickles are basically a harvesting device, no? Similar tools can be traced back around to 11,000 years ago, but normally its study is jeopardized by the poor preservation, of course. No, that's why the site of La Marmota is so important, because we have a lot of diversity in shapes, forms, and size of the different sickles. No, how are these sickles basically constituted? No, they are basically a wooden handle hafted, which to which is attached cutting edges in form or using flintstones. And what is important is they use some kind, or they manufacture some kind of resin, not probably of pine origin, in order to attach the stones to the how do you say, to the handle. No? Several studies have been going on analyzing the sickles. Taxonomic classification or identification of the handle have ruled that the most of the specimens or more of the sickles are actually made of oak wood, so genus Quercus, and also have proven, which is the most striking finding of these sickles, that pollen and phytolith remains have been preserved through the millennia in this resin. These pollen and phytoliths are believed to correspond to what actually was harvested and in the times of La Marmota. So they were found a lot of Ordeum type, Triticum type, so as you may know, barley and wheat types of plants. No, but also it gives information, or it is supposed to have preserved also remains from taxa that gives some insight on the environment. For example, these Nymphaea or Sparganium genus that are very, or genera that are very typical for these aquatic or lake environments. No. Entonces, then, what are my goals in this project? So basically, the main goal in this project is to add ancient metagenomics as a proxy to this study. No? So study basically use a new approach represented by the ancient metagenomics to the study of these sickles, and of course, successfully producing a agricultural profile of the sickles. No. During this study, or during my project, I got resin material from seven different sickles. No, sadly, or thank God, no, 4 of these actually produce or yielded DNA. It depends on the point of view. Yeah. Actually, when after the library preparation and the sequencing and the filtering by quality control, I realized that only 2 samples actually yielded useful data for the downstream analysis. For metagenomic analysis, the tool or the approach that I chose at first glance is AMETA, as we already heard of about this amazing tool. Normally, AMETA, as you know, has been developed for microbiome analysis, or like microorganism diversity. However, there is also the option to use as I did, the pre-built nucleotide or entire nucleotide database from NCBI, because I'm interested mainly on plants, in this case, but also of course on prokaryotes and everything. So I tried to catch as much as I could. Talking about the default thresholds, AMETA places these thresholds to ensure that the data that has been detected has a minimum depth of coverage, and also what we said, the breadth coverage, coverage of the genome. So this at the beginning, basically at first, I left this default threshold. Here we have the results that I got at first from Kraken Unique, which is the first step of AMETA, the first screening with AMETA. Here we see that, of course, for the first sample, no, we have a lot of bacteria and very few eukaryotic reeds. No, the main species or the most abundant taxa in this case was Aconobacter xyloxoxidans, but we also found another bacterial species from the genus of Pseudomonas or Stutzerin But also, when looking into the eukaryotic reeds, of course, we find some Homo sapiens, no? But we also were able, or the program was able to detect some fungal species that actually passed successfully or were represented after this let's say, conservative thresholds. No, among these fungi, we have the species Comatagaela fafi and the genus Malassezia. In the case of the second sample, we see that we have kind of a similar scenario, but a little bit different, so to say. No, so in this case, the greatest or the most abundant species was Comagataela fafi, which is, as we said, a fungal species. So in this case, eukaryotes are the main power, so to say, but mainly represented by these fungal species. Regarding to the bacteria, we also have again this Achromobacter xyloxoxidans, and we have several species detected of the Burkholderia cepacia complex, and also the genus Pseudomonas. What was interesting about this sample is that we were able to actually detect already in the first screening some plant species. In this case, the species that were found were Oryza sativa, no, which is the common rice, Triticum aestivum, which is the common or modern wheat, so to say. And then we have also Digitaria exilis, so another integrant or member of the family Poaceae, no, which of course is already consistent with what's already seen in the pollen analysis, no. But of course, Ameta adds this next step, which is the malt alignment. So Ameta, as you may know, builds, apart from the taking into account the output of Kraken Unique, it builds with the taxa that were detected, builds project-specific database that is built with malt and aligned again to our sample in this case. No, so here we represent basically one of basically some examples of the main species in terms of abundancy, but also the ones that whose validation also as ancient DNA appear to be more indicative of the true presence of the true ancient character of the sample. Now here again we have these Achromobacter xyloxoxidans. I have to point out that these results now correspond to only one of the samples. Because and this sample was the one who actually, as we saw before, no, it corresponds to actually this sample that has this Comagataela fafi as lead taxon. I'm still struggling a little bit because the other sample is too big, and this Ameta is very heavy program, especially the multi step. So I'm still working on that, trying to make it work. But I present you here already the results for this sample. No, again, according to the bacteria, or taking a look into the bacteria, we have this Achromobacter xyloxoxidans. With over 1 million reads, and we see this in the deamination profile. We see this increase at the end of the reads of the read to the 1 into adenine transitions, and also a general short read distribution. This pattern kind of over the analysis repeats itself also with other species of Achromobacter that appear in the analysis. The bacteria Pseudomonas aeruginosa, but also other species of Pseudomonas that appear in the analysis, and of course across different species of the Burkholderia cepacia complex, or for example the species Burkholderia lata. No? Then, according to the eukaryotic samples that we find, we see that the results are a little bit more shaky, I would say. So again, we have a good representation in terms of a number of reads. In this case, over 5 million reads were actually classified through MALT or mapped with MALT to Comagataella fafi. Here we see that the domination profile is not as stressed or as let's say indicative as the ones seen in the previous slide, and also we see a different read distribution, although they are still in the short range, no? And the same with Oryza and Triticum, we see some kind of a change or some kind of spikes in the wanting to add in the aminations, but they are not as clear, so to say, as for the bacteria, no? That's why I wanted from the from the raw Kraken unique output, I wanted to basically extract all the BDD plantae reads, because as said, if we look into the recommendations of a meta, it is said that or in the literature that these thresholds can be very, very conservative. So I wanted to take a look basically further into the species of plants that were just detected in the first analysis, no? Here we see a bunch of species, here we see a representation of the diversity, sorry. In both samples, as we see here, a comparison between the two, the plants from the family Poaceae were actually dominating the assemblage. No? Just to give some names of the most abundant species inside the assemblage, we have Brachiopodium distachyon, Aegilops tauchi, Panicum vircatum, again, the Orderum type, Triticum, Oryza species, no? Then I run map damage, and then this is where I'm a little bit also stuck, or I'm working in this discussion, because when I took the reads of Triticum and I tried to map with the so to say, with the RefSec genome, with map damage, I get these outputs, no, in this length plot and also in this cumulative deamination plot. So kind of looks, at least according to the manual, it looks kind of typical or indicative for ancient DNA. However, when we look in the that I don't show here, the misincorporation plot, we see basically a flat line. So this is a discussion that I need to work, and maybe I can get also feedback from you, no? And then we have the case of Oryza, no? Just to be clear, there were several species of Oryza that were found in this analysis. We found Oryza sativa, which is the common rice, no? And then we also found Oryza glaberrina, which is called, if I'm not mistaken, the African rice. And then basically several reads that were actually not mapped at the species level, but in the genus Oryza, no? So basically I merge all these reads and try to map them into the most complete genome that I could find, RefSec genome or reference genome that I could find. It in this case was the Oryza sativa, and I got this profile, right? Which again we see increases of domination at the ends of the reads. However, I'm also a little bit puzzled by the outcome, no? As normally I would expect that the plot, or what I have seen in the literature, that the plot is oriented just opposite, no? Basically as it is, but in the opposite. So maybe also you have any input or any input on that, it is welcome. Then, then now we come with to Comagataella fafi. again, we have a similar scenario as we see in triticum, no? When performing MAP damage, we have this barely or roughly short redistribution and this curve deamination cumulative frequencies, not? But again, not shown here, but with the misincorporation flat looks flat, no, as it would be for a modern sample, no. However, here I want to just give some discussion of some conclusions or basically the directions that I'm working to. First, the presence of species such as Achromobacter xyloxoscedans, Pseudomonas and Burkholderia, according to the literature, are consistent with soiled and agricultural environments. Basically, many species, especially Achromobacter xyloxoscedans, have been isolated from root environments, and they carry different functions. For example, involved in processes such as seed growth promotion or plant growth promotion, etc. Also, the plant assemblage with a strong representation of poaceae correlates with previous studies on phytoliths and pollen as proxies. However, it is important to say that according to the literature, classifications, especially of eukaryotic metagenomic classifications of eukaryotic organisms, must be taken with care, as normally databases are fairly incomplete, or even there is the presence of bacterial genes or bacterial genetic material mistaken by in the genomes of eukaryotic roots, no? So yeah, as said, this is the direction in which my discussion is going. And also, importantly, we have this Comagataella faffi. Comagataella faffi is actually a yeast, so a fungal species, as we say, and it has been traditionally, or was first isolated from wooden remains, in this case from the genus Quercus, or from oak, no, which was actually the raw material used for manufacturing the handling part of the sickles, no? And yeah, and basically the analysis of well, ignore this modern sickle. Sorry about that. On the ancient sickles, basically proves that the extraction protocol was able at least to produce ancient reeds, I think, and some extent, and highlights the power of preservation of the resin, no? So that was it for my side. So if you have questions, please, or inputs, I'd be happy to answer.
Thank you, Luis. Maybe to break the ice, so then people have courage afterwards. I wanted to ask you, maybe, like, I don't know if it's something that you might know, but maybe yes. What do you think made the Lake of Bracciano, like, this place be so good for preservation? Like, why do you think, or if you know, or if some colleagues have told you? So this was my first question. And then the other question was more about that if you're not scared that some of the results might be connected also to soil, that it was around, because I guess they were underwater and under soil. So if you have done some study also on the soil itself to see how was the structure.
To the first question, well, I don't know exactly the details of why is well preserved, but basically what I have heard or what I have listened, because there is very little. Let's say most of the studies in La Marmota are actually let's say archaeological in the traditional approach, so to say, no. But basically, the main conclusion is that the burial of the site was very, very fast. And what is funny about this site, to me at least, is that well, the Lake Bracciano is actually one of the most interesting parts of the lake itself in the geological point of view. It seems that it has been subject of several episodes of flooding, and then so to say, several changes in the level of the water, no? And it seems the Lake Bracciano was, or the La Marmota was fastly flooded, but not so fast that there was, so to say, like casualties, no? Because there has not been remains of human beings found in the site. And you could say, okay, but maybe they were washed somehow or they disappeared. But we find a lot of remains of cattle and animals that they have. So that makes us think that they were able, so to say, to escape. But at this point, they had to leave all these artifacts that probably would have taken a lot of time to make. So I would say it was fast, but not fatal, so to say. No?
Mm-hmm.
And the second question, yeah, of course, I am afraid that this might be soil, and I think a great proportion of the taxa that I found, of the diversity that I found, is actually soil bacteria. Right now, my main goal at this point, with the material I have and with the time I have, so to say, is basically to discuss, so to say, what is ancient, what is not, rather than to really build, so to say, at this point, a complete agricultural profile. No, that was the main goal. But on the other side, of course, we have taken some course, no, and we are also in the process of analyzing this course that will tell us more about this. No,
Thank you.
yeah.
Yes, exactly.
Yeah, hello. I have a question that is more related, kind of follow up with what Bianca asked. Because, for example, what I caught my attention was pseudomonas, because before I was working with pseudomonas, and particularly, I mean, it seems like the pseudomonas is like a very ubiquitous bacteria that will be found in sediment, will be found in water as well. So it's like I think that, I mean, I don't know if, like, how are you kind of distinguishing like this kind of possible cross contamination across? I mean, I focus on this particular genus because I know it's like everywhere basically.
Yeah, that's a very good question, and it's something that concerns me, of course. Here in this presentation, I didn't show, but of course, Ameta offers another. I mean, I can quickly show it also. So to say. So I don't know if you can see it. I seem not to be able to.
No, because I think
No.
that
No.
you're showing
no.
they presented the, I don't know,
Just
PowerPoint.
the percentage.
So maybe you have to reshare your screen.
Okay.
Okay.
Sorry, reshare here. So of course, there's also something that I realized across my samples. For example, Ameta had this scoring thing, no, and basically none of my samples kind of passed the test, no. So I'm kind of on the process now, as we see here in the edit distance. I don't have the really clean profile, or here the average nucleotide identity is not exactly as it is, although the coverage is good. So I'm kind of working in this direction. No, I think it would be important, for example, to sequence the blank that it's not done yet, and maybe move the all the reads that may have mapped into that. No. So yeah, that's also kind of the question of how could I basically discuss the data. No, for me, there are some indications that may be ancient reads in the sample. For example, these short fragmented reads. I have also to say that the deamination plot, for example, is consistent to what the library preparation, so to say. There was this I remember there was some debate also, this article about this proofreading enzymes, right? So that's why I suppose that I have only this one-sided deamination pattern, no? So yeah.
One question, and it's like not completely related, but do you know more like how depth were the artifacts like buried?
No. I just have seen like different like maps, for example, of the excavation, and they are basically just scattered. Yeah.
Yeah,
But
because
yeah.
basically the question was because also like I mean like the even if they were like covered, there is like normal like this exchange of bacteria between this like sediment from like a lake, river, anything, like across like 5-10 centimeters. So you can also like find a special bacterial species like living in the water. So if I don't know if you have access or did you like or they sample also the water. So I think not only blanks, but if you can get hands on the water data, I think will be like a great to have because like a course, water, and blanks. I think that will be like the ideal stuff if you can.
That's a great input. So that's something I would like to look into then. Yeah. Thank you.
Thank you for the presentation.
I also have some question. Can you hear me?
Yeah.
Yeah. Okay. So like you were showing this graph, if you can put the other slide, like the slide you just showed.
The slide I showed. Okay.
Yeah. I'm just wondering like because do you know how they were sequenced, these samples? NovaSeq or...
That leads to another question I have. For me, that because I said I have this deamination pattern, but actually this library construction was single-stranded, so to say, no. So as I heard, no, in the literature, actually I should see CT deaminations all the way and not GA, and that's something
so
I asked at some point, and yes, and here. For example, if I open then the PIMI, the PIMI deamination plot that also comes with a meta, kind of shows this increase, at least or what I suppose is some increase, no, in the CT, no. So I'm kind of puzzled of what is happening in this deamination plot of a meta, no. So I don't know.
Yeah, because I mean, I think if you are using NovaSeq and you have, truncated reads or shorter reads than the length you are sequencing, NovaSeq will just add a poly G. So you could see that spike as just a result of that at the end of the read.
Okay.
Yeah. And also like the what you were talking about before, the swapped plots.
Yeah.
I think that could be related to library prep, whether it is double-stranded or single-stranded.
So in this case, that was the same sample. So in theory, it was single stranded. Do you see it now? I'm sharing.
No, I don't see anything. Okay. I'm seeing.
No, we don't see it.
No. The Orisa, we see your presentation, right?
So this you meant this plot, the Orisa.
Yeah, I mean this was the swapped plot you were talking about. Yeah, this one, I mean, we had instance in which this swap depended upon the type of libraries that were sequenced for a sample. So whether it was double-stranded or single-stranded, and we were seeing kind of this thing when you had merged libraries of the same sample with mixed library prep approaches.
Yeah.
Then for the other thing, I was talking more about the slide you showed. I mean, not a slide, it was like plots from a meta. From a meta, yeah. So like that spike of G2A thingy at the end. I think that could be an artifact of if you sequenced on NovaSeq, it adds a poly G, so you will have that thing. In my opinion, I don't know, maybe it does not necessarily mean the amination.
Okay.
Yes. And yes, you showed, then I have a question, like just curiosity. You showed the sample with the human DNA in it. So I was wondering whether I don't know if you mentioned it, but there was like a tiny percentage of human.
Yeah.
And I was wondering whether you are doing something with it. Are you trying to check mitochondrial DNA or?
No, not at this point. I was not. I mean, I at some point I just extracted the human reads and passed it into the whole the genome basically, and there was no an indication that was like ancient or something somehow.
Okay, but did you include the reference with mitochondrial DNA?
I have to check then.
Because I mean, it's just
why
be
not?
Interesting because you could be seeing, okay, you don't see remains of people, but you could be seeing a signal, some passage of people,
No, that would be great. Yeah.
Thanks.
Okay, I have to check because indeed there was the sequencing was actually with a NovaSeq machine indeed. However, when I ran this fast QC in the quality control, I couldn't see any overrepresented how do you say? Yeah, overrepresented sequences, no? So, but it's something that I have to look into.
Yeah, I mean, to me, on the edit distance is strong indicator that something is going on there.
Yeah.
Thank you. But really, very interesting.
Yeah, I would also check. Sorry, Andreas, is your... Yeah,
yes,
sorry.
Yes,
No, go.
Thanks for a great talk. I was just wondering, in regards to the two samples you got that had DNA in them or worked, could you see any similarities between the material? Like, was there anything, is there like a giveaway or something you can look for in the future for these materials that yield DNA in comparison to the ones that don't?
The only difference, that's a cat. Sorry. The only difference I could see was actually the as you see in this. Are you seeing my presentation?
No, now we see the...
No. Okay. That's technology. Okay. And now probably.
Yes, now yes.
Yeah. The only thing I could say, so basically the sample, one of the samples was this, and the other sample is the 6 that is this, and is the differences on where it was basically sampled. No, if it was in the surface of the sickle or in the interior part. For the LM4, as it was a sample that I received later, I don't have that information. I have the information from the other ones. For example, we see that here the 3 and the 5 that didn't work were coming from more inside the sickle, so to say, and the best sample was the 6 that come from the surface. But for example, this other one that didn't work was also a surface sample, so to say, So I couldn't really find at this point like a strong correlation, to say, But yeah.
There's also a few samples, but I was just wondering. Thanks.
Yeah. Thank you.
No, mine was just a comment that I was thinking that I don't know, well, if you have checked, but maybe yes, you have the little book of smiley plots, because there are some of them that might be worth checking. Either there is this barcode ligation bias, or also checking what Maria said.
Yeah. That in the end, like for my discussion, I kind of, because of I knew there was something off in this ameta, so to say, thing, no.
Mm-hmm.
So that's why I don't know what you think, for example, of this PMD plot can be also like an artifact of some sequencing or something like that.
We don't see it.
I don't see.
Sorry.
Here, yeah.
Yes, I'm thinking. Yeah, it could be something like this. At least it's very similar to the picture that they show about the internal barcode ligation bias. But yeah, so that's something I think you could look into that, and and the cases that also Maria showed.
I also was thinking that there is a lot of in this case you don't see the G2A thing, but like there's also, like, to me, a lot of noise, no? I don't know, like a lot of other things that yeah. So it kind of leaves me a little bit yeah. But yeah, there are things that I have to, of course, look into.
Yes, Minna.
Yeah. Hey, Luis, thanks for the great presentation.
Sure.
Yeah, so I was thinking if you were interested in plants, so maybe there is some database only for plants.
Mm-hmm.
And I was thinking, are you considering that you could try to check your sequencing reads against that plant database and then compare the results with AMETA if you get more like more taxonomy from plants?
Yeah. That was one of the steps that I wanted to do. I'm also worried all the time to keep always like the mapping as competitive as possible. So I was thinking of doing something like I don't know, like for example, extracting the reads from Kraken and then building a database. I don't know, even with something like Boti or something, and then try to run map damage, and that is basically what I did for producing these plots. So basically, that I think I forgot to mention. Then I extracted the reads from Kraken, and then I basically a database of RegSeq or reference sequences of of all the Viridae plantae, or basically huge database. I have to split it into I don't know, 1 million parts, but in the end I mapped them with Boti, and then I applied this I don't know, this software NGSLCA for trying to reproduce what Ameta does or Mal does with this lowest common ancestor thing, and then I got these things, no. These plots, no, and then of course happens. For example, the Oryza is the result of that, of mixing all the samples that all the reads that has something to do with Oryza, the genus and everything. But I have this deal with Triticum, no, for example, no, like shorter reads, curved but flat. So then I remember I asked in the Not Stupid Questions forum. And Nikolai, for example, asked he says that it looks that the reads are ancient, but the reference is wrong or something, like it's aligned to the wrong reference. I don't know how to make sense of that, so to say, but yeah.
Yeah, I think the reference is a good. It's a good thing to start troubleshooting, I guess, because I was thinking just like my next question was like if you had tried different reference genomes.
Yeah. For example, I tried, or I was trying. I haven't done it yet, but I wanted to do the same as I did with the rice, the same with triticum. Like I have also, because in the end I was thinking that for example the genome of wheat, as far as I know at least, is kind of a young genome, no? So basically probably what they had in the Neolithic was looking different. No, I read about this so to say, this origin from this Taushi, this other grain, or like the wild type Triticum urartu. I always hit this rock a little bit, in which like the of course the most complete genomes are the ones for like the commercial ones, so to say, for the Triticum. I don't know, as far as I look into, but yeah, one option would be to put all of them again together. No, I don't know. Maybe that would be a way to go, but yeah.
Yeah, we're trying to do, I don't know if they exist, but like to make some genome assembly from public data, like long reads, because if I'm not wrong, there should be something on the Triticum, but I'm not 100% confident, although I'm almost sure.
Yeah, what I have seen is from barley. There is something from barley. So that I haven't done it yet, but that I had it in mind to download the genome they have and see, basically, no. But yes, lots of things to do.
Yeah, because also your edit distance seems to say something like this, that it might be not the perfect reference genome. So I don't know, of course, the edit distance. Edit distance. So I don't know if, of course, like I guess to do de novo assembly is very complicated with those short reads.
Yeah.
Or yeah.
I mean, I don't know if this is something that people do looking into the chloroplast.
Yes, I think so.
I think there is some years ago I used some software called Novoplasty that is do you know that is specific for chloroplast assembly, no? And this is what they use in well, I don't know. Yeah, basically I heard about this, or I learned it in the university, no? But I don't know how it performs for ancient data, no? Because that's the thing. The sample LM004, that was the one with the spikes, the GA spikes, was the one who was like single stranded. Also had this proof reading enzyme, or I don't know. In the end, I think the debate was just skipping, no, the deaminated positions. But for example, the sixth had a good quality, and then it should be like is a normal true sec kit, no? But yeah, so there was also something in this kit they used in the 4 sample about like filtering out shorter reads. And in general, when I did this analysis also on this 4 sample, I get also a higher read distribution, longer read distribution than the one in the 6. No, so the 4 is a little bit then difficult to authenticate. It's been a little bit difficult, but yeah.
How old is that one sickle that gives a lot of that gave the data, or had the good eukaryotic data?
So they are all, I mean, the period of occupation of La Marmota was the 6 millennia before Christ. So they all are in a window of 400 years. That was the occupation. So
okay. I was just wondering in the sense of how much damage you would expect.
Yeah, there are also some studies in which the resin kind of, or there was a study in which the resin kind of at some point could shield the reads I read, but I don't know how is if this is the case or not, but something also to discuss. No?
Does someone has other question for Luis? Okay, if not, I'm gonna stop.